with two of our heavyweight, if you will, laboratory manager, director people, Dr. Melanie Oakes, who is in charge of the Genomics high throughput facility, and Dr. Grant McGregor, who's in charge of the Transgenic mouse facility. And I'll just mention on behalf of the Genomics facility, that Jenny Wu, who's in the back row there, raising her hand briefly, is our bioinformatics director in the Genomics facility, also a recharge feature of that facility. So without further ado, I think we'll just get going. Melanie has the clever title of long and short of sequencing, and I'll let her explain what that means.
Well, happy Monday, everybody. It's really my pleasure to be able to come and to spend a few minutes talking to you about the work we do in the Genomic high throughput facility here on campus. I've entitled it The Long and Short of Sequencing, mostly because we have long reads and short reads, and so that is something that I'll explain as we progress. The mission that we have in our Genomic high throughput facility is to bring emerging nucleic acid technologies to UCI. And we want to facilitate their use by providing services. We do workshops as well as technology talks. And the way that we do this is that we, for instance, if we want to look at our description, is that we provide genome-wide analysis for clients, that would be you, that would be interested in gene expression as well as regulation of gene expression and genome and sequence variation. So the actual instrumentation and equipment that we have that's available in Sprague Hall right here on the School of Medicine side of campus is the microarray, and I'll be honest that most of our microarray clients have now transitioned to RNA-seq on the Illumina sequencers. We have NanoString, which was an addition that was through Dr. Mortazavi here on campus. And then the Illumina platforms we have, this year we added the MySeq in April, and that was from a grant that Dr. Ilham Masudi submitted to the Office of Research, and other campus organizations participated in contributions to that, like the Microbiome Initiative, School of Pharm Sci, School of Physical Sciences. So we're grateful to have that now on campus as a service. We also have the HiSeq 2500 and the HiSeq 4000. This year we also exchanged the PacBio RS2 sequencer we had to the Sequel, and again, I'm really appreciative to the campus for helping us do that. PacBio came and offered us actually I don't want to call it a rebate because that's not the right word, but they were willing to take our RS2 and give us some compensation back towards the Sequel purchase. We also have some single cell platforms, and I'll explain these more in depth as my talk progresses, but we have the Fluidigm C1, and we also have the Chromium droplet GEMCO technology. It's not an actual sequencing platform, but we have what's called optical mapping for very long reads, and this is the BioNano Iris platform. In terms of bioinformatics, we offer support in terms of the design for high throughput studies. We provide assistance with grants. We write letters of support for your grant proposals. And we do custom analysis, we meaning mostly Dr. Jenny Wu. And we also have some software programs available on campus, the CLC Bio Workbench, as well as the Ingenuity Pathway Analysis programs. So if we talk about long reads in our facility, we're talking mostly about PacBio and the Sequel, and we're talking about 20 kb lengths of sequence, even up to 40 kb if it's an amplicon. Sometimes it can even get up to 60 kb if the polymerase is really happy on your template. For short reads, we're talking about things that, if I say it's 2 by 300, I'm talking about paired in, where if we have a length of a DNA fragment, we're going 300 bases from one end and sequencing 300 bases from the opposite end. So the total would be 600 bases on your length of fragment. So the 2 by 300 is possible on the MySeq. That's what the MySeq has brought to this campus now. On the 2500, we can do up to 2 by 250. And on the 4000 instrument, we can do 2 by 150. So what does that mean for us? You know, why would we want to choose short reads? Why would we want to choose long reads? And really short reads have a wide range of applications that that particular technology is really useful for. So if we're talking about RNA, we can look at splice isosome abundance. We can look at gene expression, differential gene expression. We can look at small RNA. And if we're talking about DNA, we can do ChIP-seq, we can do methyl-seq, we can do exome sequencing, we can do even metagenomics rearrangements. So how does that compare with the long-read sequencing? So with the long-read sequencing, it still has some of those components. For instance, genome resequencing, we can look at rearrangements now because it's over a longer length of fragment. We can look at DNA methylation, de novo sequencing, and for the RNA, we can also look at different splice isoform variants. So really, these are complementary approaches. In one hand, on the Illumina platform, and I'll show you how the actual sequencing chemistry works, we're looking at still images that have been taken by a camera of clusters. And these clusters are approximately 1,000 clonal copies of the same template. We can have, like I've mentioned, it's short read up to 2 by 300. The interesting part, if we're talking about DNA repeats, for instance, repeats are not analyzable on this particular platform because it's short. So that would be a platform, for instance, if you were interested in repeats, that we would want to go to the PacBio system. It does have very high accuracy, so the Illumina platform's less than 0.5% accurate. There is considerable base bias, and this is because there's PCR amplification steps involved in this technology. There's PCR when you're making the library, potentially. There's also PCR on the actual chemistry, on the sequencing itself, and I'll also show you where that happens. You can get de novo assemblies of thousands of scaffolds with this particular platform. Now, the PacBio is a little different, and that's actually a movie recording. And I'll talk a little bit about that when we get to the PacBio platform. But it's a movie recording of single molecule fluorescence. And like I mentioned, it can go up to 60 kilobase pairs with N50s, about 23 kb, which is pretty long. Because it is got these long and up to 60 kb, we can actually span retro elements, and so it can be very good for repetitive regions if that's something that you're really interested in. We can get up to about 20 gigabases per SMRT cell now with the new sequencer. The error rate is 15%, and that sounds a little ominous, but in a little bit I'll show you how some of that can actually increase in quality because of the way that the system is built with the SMRT Bell. In terms of affordability, it's about 3 times higher than the Illumina platform, and you can get near perfect genome assemblies. So bottom line is you need to know when to go long and when to stay short. So the PacBio platform, and this is what our new sequel looks like, is that it has 1 million of what they call these zero mode waveguides, or ZMWs. And each one of them is about 70 nanometers in diameter. At the bottom of this surface is polymerase gets attached to the bottom, and it's basically through an avidin. The polymerase is actually biotinylated, and there's avidin on the surface. So once you have a template, which would be, this is your DNA template right here, and here's the polymerase that's bound. It sits on the bottom, and then there's a laser that excites into the active site of that polymerase. Up here floating around are fluorescent nucleotides, so that when the complementary nucleotide comes into the active site, then it can see what fluorescence that is. So then over here, you can see, here's a visual graphic of the polymerase, is that when it comes into the active site, the camera can read that. What's really interesting is that how long it stays in that active site and what the fluorescence is actually dependent on which nucleotide it is. So the instrument's looking not only at what fluorescence is in there, but also the amount of time that it's in there. And that's how it's able to do the sequencing. They've also engineered the polymerase to be a little slower. So it adds about 2 to 3 nucleotides per second, depending on which nucleotide is actually being added in. So what is the workflow for this particular platform? If we start with a very long genomic DNA fragment, we do a fragmentation step. And again, because it's a long fragment, we want them really big. So we like the fragments to be about 48 kb and even higher. And then the ends are repaired, and then hairpin loop adapters are ligated onto each end. This is where they get the smart bell word, okay? This then gets purified, and when it gets ready for sequencing, then it binds, we anneal a primer, we bind the polymerase, and then it goes on to the sequencer. So the way that the polymerase reads through it is that the polymerase would start at this one end of the smart bell. It would go through in one direction, goes around this hairpin, and then goes back on the other side. Depending on how long it is or how short it is, it can actually cycle through many times this one particular sequence. So what happens in the polymerase read is that you would have these, you, for instance, would have this purple right here, which is here. Then it reads through the hairpin loop, which is green. And then you have the complementary strand, which is now in yellow. So you can actually, and these are called subreads. So these subreads can actually stack up. And if you recall, I mentioned that the error rate's about 15%, but that error rate is random. So every time you resequence one of these strands, then it compensates and it gets error corrected. So you can actually end up with pretty high accuracy depending on how many subreads you get. There's also a workflow for ISOseek, which is an RNA isoform workflow. Starting from total RNA, you would make a full-length first-strand cDNA, make amplified cDNA, and then sometimes people do a size selection. The reason why is if you think about those 70 nanometer zero-mode waveguides, that if you have templates in there that are vastly different sizes, there's going to be a bias for the shorter ones to get in first compared to the longer ones. So a lot of times people will go ahead and do a size selection so that when you sequence them, you're giving all of them equal opportunity in order to be sequenced. So if we were to look at why is this particular platform so helpful for ISOseek, if you have an example here, this would be where we have potentially 5 exons. That 5 exons, if you have an isoform that's got all 5, it's going to be this length, right? And if you have them where it only contains exons 2, 3, 4, and 5, it's going to be a little bit shorter, 1, 3, 5, and whatever combinations. So this has really been a usable tool, and here on campus, Dr. Mortazavi is involved in an ENCODE project looking at RNA isoforms, and we're doing presently a lot of sequencing for him using this particular platform called ISOseek. So moving on to my short technology, most of you have heard the name Illumina. Some of you may have even been clients, and we've done Illumina sequencing for you, and so you may be very familiar with the workflow, but it's a sample preparation, a quantification, a clustering, a sequencing, and then analysis type workflow. And the bottom line for Illumina, which is really kind of nice, is that for this short read technology, if you can put adapters on it, then we can sequence it. So there's been a lot of different applications, even here on campus, that have been developed using this particular technology. Hi-C, ChIA-PET, these would be both conformational information about chromatin. For RNA, it's RNA-seq as well as riboseq, small RNA, MDIP-seq, lots of different applications that have been developed for this platform. So the workflow, as I mentioned, is library preparation. And what that means is that we're going to need some type of DNA strand that has Illumina-specific oligo sequences on either end, and we call those adapters. And then on the flow cell, we're going to do what's called clustering, and this is a PCR amplification step that is actually on the platform. And then we're going to sequence. The sequencing is really kind of taken off of the original Solexa sequencing, and that a primer binds to all of those templates in this cluster, and then it adds one fluorescent nucleotide at a time. So what happens is that what it's called cycle 1, it adds one base, and then it images all of the clusters on the flow cell. Afterwards, the fluorescence is cut off and it adds the next base, so that in a 4-channel system, which meaning that all 4 nucleotides have 4 different fluorescents, then for instance here in 1, this would be a T, and then in 2, it would be a G, cycle 3 is a C, and so on. In addition to 4-channel, there's also a 2-channel technology for Illumina. This would be your NextSeqs, and this would also be, for instance, the new NovaSeqs that have just come out. And in this one, the way that they're making it much, much faster is that it's just 2 colors, so that T and C are green and red, and then A is a mixture of green and red signals, and G is no signal at all. So in terms of the clustering, I'm showing you an example in this image as to what a flow cell looks like. Typically, they have 8 lanes, and on the surface, on the bottom, as well as the top, is a lawn of these oligonucleotides. And this is why we need the adapters put on. So from these oligonucleotides, then your template will bind, and it will use this oligonucleotide as a primer and make a complementary strand. So our new ISOseek 4000, I've talked about this lawn of oligos, right? So typically on the older instruments, and even on the MySeq and on the 2500 we have, on the NextSeq, it has random oligos on the flow cell. And what the new innovation was in coming up to the ISOseek 4000 as well as to the NovaSeq is this patterned flow cell. And the patterned flow cell lets us have a defined lawn across the entire flow cell. So for the ISOseek 4000, we can have 478 million of these nanowells on 1 lane. It allows for optimized feature cluster spacing, simplified imaging. It has made things much faster. So what are some of the other applications that we have for the that we have in our facility that are for the short read sequencing? And one of them is the 10x Genomics single cell, which is also referred to or described as DropSeq. And this one utilizes something called a Gem-Code bead technology. And that's the big bead that you see here in the middle of this slide. And what each bead has is primers that are all around the outside of this bead. And for each bead, they're going to have one 10x barcode here, which is going to be a cell-specific barcode. Then it's going to have a unique molecular index, and then it's going to have a poly-T region. So what happens in this technology, and it's basically just a little box and it's a vacuum pump, is that oil comes into it as well as the Gem-Code beads and the cells. So people prepare a cell suspension that would be single cells. So within the oil droplet, you hope to get one Gem-Code bead and one cell. Then what happens is that within the droplet, the Gem-Code bead dissolves and releases all of its primers, and then the cell lyses and releases all of its messenger RNA. So now those oligo-DT regions are able to bind to your messenger RNA, and it creates a complementary strand. So that strand now has the 3' UTR region of an mRNA. It has a unique molecular identifier, and then it has a cell-specific barcode, because each one droplet should have one bead and one cell. So then the platform, or 10x Genomics, has a software program that's available. It's here on HPC. You put your data into what's called Cell Ranger, and it actually tells you how many single cells did you get. And we can get, or at least right now, the specifications are up to 10,000 single cells. So the other technology we have, the older one, is the Fluidigm single cell C1 capture. That one has a small, it's a microfluidic capture system that can capture up to 96 single cells. What makes it different from the DropSeq is that for the 96, you can get whole transcript rather than just the 3' UTR region. So some people still prefer a whole transcript approach, and you can do it with the Fluidigm single cell. So I call this one coming along and going long, and this is called Nanopore. We have a Min ion in our lab, and there are other Min ions here on campus, and it's definitely becoming a more robust technology. It's got a protein molecule, hemolysin, and what happens in this is that there is a electrically resistant, I'm trying to see if I can see it. It's electrically resistant across on either side of that protein. And then using an ionic current, it flows through the pore in that protein, and you now have a voltage that's between or that's on the opposite sides of that silicon. So as a DNA is coming through, it interrupts the voltage, disrupts the current, and so you can get a reading as to what that base is. So some of the advantages of this is that, you know, for the initial adoption program for using this, or what they called early access, for $1,000, you could get one of these little Min ions. They have about 512 of these nanochannels and about 10 to 20 gigabases of information. It's highly error prone, similar to the PacBio system, but it is cheap, it's fast, and it's scalable. And it now can be very efficient. So the Min ion can grow to the ProMeth ion, where they can put about 48 of these flow cells into this particular device. So now that you have approximately 3,000 of these nanochannels each, with now the potential for terabases of information coming out. What's going to happen in the sequel? Well, the roadmap for the sequel, we already have 1 million ZMWs on a SMRT Cell, but supposedly within the next year or year and a half, it will go up to 8 million. So definitely, sequel is feeling the pinch from Oxford Nanopore. And so you have kind of a robust competition going on at this point. And in terms of optical mapping, what we have in our lab right now is the IRIS system, but Bionano has come out with a new platform called the Sapphire. And it will do optical mapping from 100,000 base pairs up to mega base pair lengths, and approximately 640 gigabases of throughput on a chip. So you can do a human genome now on one chip. Optical mapping is a system where, again, you're taking images, but what they've done is that they've been able to label the DNA in a sequence-specific way. And the new technology they have on the Sapphire is that they're not having to nick the DNA to label it with a fluorescent tag. It can do it without having to nick. So you get these long lengths of sequence with these fluorescent tags that have labeled it. And it's great for repeats. It's great for the ends of telomeres, for instance. So there's also very high throughput for the short sequencing as well. So the NovaSeq 6000 has come out. It can do up to 3 terabases in a run. It can have 9 billion clusters and do up to 2 by 150. So and they're doing this with scalability. So what they've been doing is introducing various versions of their flow cell. The S4 is the one that's 3 terabases, and then they have the S2, the S1, and they've just now come out with a new one that is basically 125 gigabases on a lane. The sequencing, as I mentioned before, is a 2-channel. So T would be the green laser, C the red laser, and then A is represented as a combination of both, and the G is the absence of any fluorescence. So in terms of talking about the how much things cost, obviously with the bigger output machines, the price per gigabase dramatically reduces. So the more your instrument costs, the less the sequencing costs your clients. Our HiSeq 4000 is still competitive with these lower output or lower throughput flow cells, but when it gets to the S4, the S4 definitely is the cheapest per gigabase right now. So, and more. So what's coming? We just finished a boot camp for a technology called RNAscope. So RNAscope, because we're doing so much single-cell sequencing right now, the idea is people are identifying these subpopulations, and now we want to get them back into what does it look like in the context of the original tissue. So RNAscope is sort of a basic technique to do that in terms of that it can look at up to 4 different RNAs in a tissue slice. Next year, supposedly, it will be able to do 12. So what it will do is different iterations. And I want to put out a thanks to Adila, who helped with our workshop. We really appreciated how quickly. So in basically 1 hour, she could look at 9 different slides. Adila, I'm looking for you. Yes? 9 different slides? And be able to show beautiful pictures. So we really appreciated her help with that boot camp. The other thing we'd like to do is single-cell ATAC-seq, which is looking at chromatin accessibility, and single-cell RNA-seq in the same cell. So there's a recent publication that just came out, and we're looking to build that into our genomics core, is being able to say, hey, this is the gene expression, this is the chromatin accessibility, and basically be able to tie that back to a single cell, which would be awesome. Some of the other things that we are seeing develop, there's now a single-cell RNA nanopore sequencing that a group at UC Santa Cruz has developed. It's certainly something here that we could try on some of our Oxford MinION products. The other thing that people have been telling me, or researchers have been telling me they're interested in, is looking at proteins. And so there's another new platform that's come out called an Ion Path that would image approximately 40 proteins on a single slide. So there's lots of new technologies coming out, and I am open to what kind of things do you want to see and would you like to have here at UCI. We have a website, and on our website is our order forms as well as some guidelines. We also list publications. So and that's me going through PubMed, right, trying to locate all of you. So if there's something you've published and you don't see it and you've used our facility, please let me know, and I would love to put it on our website. Also is the URL for our website there. In terms of contacts, don't contact Suzanne, she's too busy, but contact me, unless you want to complain about me to Suzanne, then you could do that. We also have staff in the core that are more than happy, and we have a facility lab phone that you're welcome to call. Jenny and I love to meet with people to discuss their projects. In fact, if you're planning something to do, that would be our preference, so that we could have an idea of what your project is that you want to work on. And with Jenny involved, we can start from the very beginning all the way to the very end on the analysis and really assist you with that. I also want to make sure and give thanks to IGB. They're the ones that all of our sequences are transferred over to IGB, and they do the preparation in order to send them out to you as links and for being able to download. And I'd also like to thank Harry Mangalam, who's always available to help us with our IT needs. And with that, I'm happy to answer any questions. Thank you. Yes. Hi, Kim.
So the ion path for protein sequence, would that take care of protein that have modification?
I don't know. It's just something that's new come out, and we're looking for a platform to bring in, especially for the single cell analysis part of our core. And so that one has been recommended. There's also another one that we're looking at as well. But the idea is being able to localize a lot of proteins on single cells.
Say a little more, Melanie, about the single cell ATAC-seq. That sounds like a very powerful.
It is, and it's actually really clever. The way that they do it is just done in multi-well plates. And so, so you do a step where you then isolate the nuclei. So you kind of break open the cell, you sort of pellet the nuclei so it stays at the bottom, and then you take the soup for all your mRNA. And so it sounds very simple, and theoretically it should work, and certainly in that paper they've described a technique, and we sure want to do it. Yeah.
Yeah, let me know when it's ready.
Okay. Brandon, question?
No, ma'am.
Yes.
Yes, question about, you mentioned that you do the analysis also, is that?
We do.
You do that, or is that somebody else that uses it?
Jenny, would you like to address that?
Definitely. So we're here, we've been here for almost 10 years. So we do provide sequencing data analysis as well as statistics data analysis. So especially for RNA-seq whole genome sequencing or whole exome sequencing and recently single cell analysis. So depending on your analysis needs, we're happy to discuss with you as to your specific analysis of project. So we're on a recharge basis.
Right.
And I'll just add a special plug there. We also have workshops. And so a lot of the laboratories have found that coming in through the students and postdocs being involved in workshops and getting a grounding in where resources are available for doing analysis is a good start to the relationship to do that. But if you want to fully put it on the facility, that's available as a recharge. But they also run workshops, and those are roughly quarterly, but they're really on an as-demand basis. So if you contact Jenny or Melanie, we can set up workshops when we get a critical number of people. So,
Speaking of workshops, we're going to have one at the end of this month on the 25th, and it will be an afternoon mini symposium that is in combination of 10x, Militini, who helps with sample prep, as well as Illumina. So I'll be sending out email. We do need some registration, but that'll be coming out. Hopefully I'll get to it today. And it's also available on our website, so I will be putting that information on our website. Then in October, we're going to have a sequel launch party. And so that will be again in the afternoon, and we'll have speakers that will be here at UCI. Dr. Mortazavi is going to be talking about his ISOseek project on the sequel. And then they'll also be telling you about new chemistry that's coming along. That's a full afternoon, and they are calling it a party. And so, transition, okay. Thank you.
Okay, good afternoon. So I'm Grant McGregor. I'm the current scientific director of the Transgenic mouse facility on campus, which is also known as the Mouse Genome Engineering Facility. What I'd like to do today really is emphasize this: what can we do for you? Because we're a core service. So I'd like to spend the next 25 minutes or so giving you a little bit of background on why the mouse is so important as a way to model human development, homeostasis, and disease. And then I'd like to introduce you to the staff of the TMF, give you an idea of what we do and who we're supported by. Brandon, thank you. Suzanne. Pramod. Finish up giving you a couple of examples to sort of stimulate you to think about how you might be able to use CRISPR in your research programs to try and make novel engineered mice that you could use to address a particular experimental question. Okay, so many of us are funded by the NIH. NIH's core mission is to try and understand and treat human diseases. So for over a century now, the mouse has been used as a model genetic system to understand human development and disease. In the past 60 years or so, really, it was with the advent of the development of in vitro culture systems for mouse pre-implantation embryos that John Biggers and McLaren and various other people developed that allowed us to manipulate the mouse embryo genetically and then to transfer that back into pseudo-pregnant females to obtain live offspring that were genetically modified. So for the past 50 years or so then, this has been used to manipulate the mouse genome to try and model what happens and understand what happens in human genetic disease and human development and homeostasis. So there is a very rich history of knowledge of mouse physiology and genetics, again, about 100 years worth of this, that we can leverage to try and model what happens in humans. As Melanie's introduced to you, there has been, to me, truly dizzying advances in genetic sequencing and mapping. It's incredible how quickly these new technologies develop. When you combine that with in vitro culture of embryos, with the ability with stem cell technology, homologous recombination, and then more recently with CRISPR-Cas9 type technology, these powerful tools then really allow you to manipulate the mouse with precision down to the base pair. Okay, so take the impact of sequencing, etc., on the human medical environment, and personalized genomic medicine, which I'm sure you've all heard about, is now becoming a reality. And so it's not out of the realm of possibility that people will show up in a clinic and in the not too distant future will say, well, we're not quite sure how that happened, but let's make a mouse model and try and understand the basis for your particular developmental or homeostatic abnormality. So our mission statement here in the TMF or the MGEF really is to enable use of the mouse as a model genetic system to investigate the mechanisms of mammalian development, homeostasis, and disease. Okay, that's what we do. All right, so if you go to our homepage, it's easy. You just Google TMF and UCI, and it'll take you to the homepage here. We are located under the resources of EULAR. They provide us with a lot of infrastructure support without which we couldn't operate. And EULAR of research. Probably the two most important. If you have a question, email us. If you click this link, it will link to John Newman, who's our managing director, who's here in the front. And John is the recipient of all these emails then that says, I have a question about, and either John or myself will try and address that for you. The other link that's particularly useful here is online resources. If you click on this, you'll actually find a huge page that lists genetic resources, physiologic and anatomical resources, all to do with the mouse. There are myriad things here that you may find will pique your interest. Okay, so that's how to get hold of us. Who are we? Well, as I mentioned already, John Newman, who's down here in the front, is our managing director who holds the whole ship together. He's the initial point of contact for projects. If you need something, we have a question link, it will go to John. Shimako Kawauchi has joined us recently. Shimako is actually covering mostly a large U54 program that we're involved in to make new models of late onset Alzheimer's disease. In the staff research associates, we are fortunate to have Dr. Kaixuan Shi, who's our surgeon. Literally, he microinjects and does embryo transfers and is involved in IVF and cryopreservation. He does a fabulous job. Shuling Wang is our highly talented embryonic stem cell culture person who also helps design your projects for doing ES cell-based targeting. And she does an awful lot of molecular biology to try and keep up with the modifications that we're doing in the core as well. And down in the animal room, Sherry Collins here does yeoman's work, really, in terms of managing all the colonies. And when you're getting your animals delivered on campus here, Sherry's the point of contact, will make sure you get the right mice with the right genotypes. And then I basically try and stay out of their way. These guys work remarkably hard and are remarkably good at working with each other, and we're very lucky to have them here on campus. And on Friday, I actually did a back of the envelope calculation, and between the 6 of us, we have over 130 years of experience in working with genetically modified mice. Okay, and that's something that is to your advantage, I would suggest. So my good friend Rahul Warrior here said to me on Friday, why should we use your TMF? Okay, and I thought, okay, well, let's talk about that. Many of you will have seen that there are myriad companies now, Charles River, Applied Stem Cell, Cyogen, who are very good at sticking posters up all around campus here, Taconic, etc. So there's an awful lot of other places out there that you could go and get your mouse models made. So why should you come to the TMF? Well, you don't have to, but here's why I would suggest it's good for you. Again, expertise. We have about 130 years of experience. This means that we are familiar with the common pitfalls involved with these sort of designs. And we would implore you to contact us at the outset, kind of like the GHTF. Contact us early when you have an idea that you want to try and modify the mouse genome, and let us work with you and try and partner so we can get you the fastest and most effective approach. Hopefully you will find that we're service-oriented. We have short response time, the most rapid turnaround possible for your project, and we deliver very high quality of services. We get you the leading edge technology. So the Office of Research is nice enough to sponsor people within the TMF to go to scientific meetings. That's where they pick up on emerging technologies, and we try and port those where necessary into the TMF. We will work with you to develop custom services. Campus that say, oh, I'd like to try this, but nobody seems to sell it. And they say, well, we'll do it for you. And we'll work out the details of any cost. And what that means on average is that we can undercut any commercial outfit, even the ones that are based in China, such as Cyogen, for example. So a colleague in School of Medicine recently paid $60,000 for a new mouse model with a humanized allele. A bit later, maybe. The defined health status. Mice coming from China can have questionable health statuses. This is something our veterinarians are very aware of here. So we make mice for all investigators at UCLA, and when they go up there, they go straight into their animal rooms because they trust our QC. And then we can do all sorts of other things, such as making new embryos. Just a relatively short list of reasons why I would encourage you to use us, or at least to consider using us. So a lot of people in the US have done that. Many of you may think that we make mice for investigators on campus, but here's a list of places in the US that are clients of ours, either single use so far or repeat use clients. And again, all of this is possible because of support from the Office of Research through your indirect costs, from the School of Bioscience, School of Medicine, and from a couple of large core grants, one from the Cancer Center, and another more recent one here from what's called Model AD, which is this U54 to make mouse models of late onset Alzheimer's disease. Okay, so what are the sort of services that we can provide? The first thing is we will give you advice. If you email us and say, I have a question, this may upset Suzanne, but we actually do this for the most part. We try and do it free if it's a quick and easy answer. I'm sorry, okay? If it is something that requires considerable amount of thought and considerable amount of planning, then yes, we will try and recharge you, but we try and keep our rates as low as possible. Like the GHTF, we'll give you letters of support, we'll give you descriptions of methods, resources, we will develop a resource sheet for you to add to your grants, animal sharing plans, all these sorts of things. Just email us and we'll give those to you. Some of these are also available on the Cancer Research Institute's website. Marion Waterman did a wonderful job of assembling a lot of those in that location. Probably only over 70% of our work now is CRISPR-based. So if it can be CRISPR, we'll try and do it for you. If you want to make knockouts in cell lines, we won't do that for you, but we will give you the counseling and technical expertise how to do it, and we'll try and help you with equipment that will allow you to analyze your cell lines. So the barrier we're trying to reduce then, in the absence of a CRISPR cell line core, we're trying to sort of step in there and help you get to where you want to be. We'll make small transgene and back either random integration or targeted integration mice. We'll make you ES-cell-derived mice from the projects or even using ES-cells in-house. We will partner with you to develop ad hoc methods. If you have some idea that you want to try and develop, we'll work with you. We'll source animals for you. That's the first question when somebody says, I want to make a knockout of this gene. as John desperately rushes through all the databases and says, oh, can I find one that's got this so we don't have to make it? Okay, it's just because we're so busy. And again, we'll give you assistance with methods and results sections for manuscripts, plus a lot more. So literally and figuratively, the bottom line here is if you have a question, email us, okay? And we'll get back to you as soon as we can, and hopefully we'll be able to help you. Just in passing, I'd mention an instrument that the Cancer Center helped us acquire. This is a fragment analyzer, and the beauty of this instrument is that it's capillary-based. It works with 96-well trays, and it gives you pretty high resolution in terms of sizing DNA. So if you did want to, for example, do a screen in a cell line of your choice to look for knockouts in particular genes, this is a nice instrument because you can bring down your plates, we'll load them up and run the analysis for you, and then tell you which clones have particular alterations in alleles. So this isn't advertised yet on the website, but if you're interested in using this, email John and he can let you know what the recharge is, which is really low. Okay, so there I was sitting in the audience looking at all the stuff Melanie presented, thinking, oh God, you know, I'm so out of touch with DNA sequencing, which I really am. But I thought, well, I shouldn't anticipate that everybody here knows how CRISPR and Cas9, et cetera, works. So what I'd like to do is give you a little bit of background on this, and then show you a couple of examples of what we do within the core that uses this type of technology. So this of this enzyme is that it in an RNA-dependent manner identify a unique location within the genome, and it will make a double-strand break in that location. What it needs to do this is a triplet nucleotide sequence. For the case of Cas9, this is any base, although some are preferred, and then a G and another G. And in combination with this CRISPR RNA, which recognizes 20 bases of homology, sometimes it can recognize less, the enzyme will then make a double-stranded break between the third and fourth nucleotide downstream here from this PAM site, okay? So if you want to make a double-strand break or mutate a gene, then really all you need to do is find a PAM site within your gene of interest and to make sure that it isn't going to cut somewhere else that might confound your experiment. And of course, there are nice websites. There are lots of these now, but this is one of the original ones at MIT. And so what you do is to plug in a search term, in this case, a few hundred bases of a region that we'd like to try and make a double-strand break. And the search term. And the algorithm will then tell you which one is best based on off-target location. So this is, in general, the sort of strategy that John or I would use, mostly John, where we're trying to identify guides. Once we identify the guides, and what we do is to get CRISPR RNAs and transactivating CRISPR RNAs, track RNA made. We get these made by IDT. They're a very, very good company, and their quality control is very, very good. What we'll do then is mix these together with Cas9 protein, and there are a variety of variants with Cas9 now that have low off-target effects. And this ribonucleoprotein, which is brew, we then microinject into surface. And these then can either be cultured to blastocysts and analyzed molecularly, or more often than not, you could go analyze conundrum, but based on 50 years or so of molecular and at the top here, it's going to be cut by Cas9, and here's your double-strand break. Double-strand breaks are very potent in eukaryotic systems, and they send a signal that says, quick, fix me. Cells really don't like these. The first process usually involves binding of a variety of proteins, which I haven't indicated here to convey the signal. But for simplicity here, what you get is resection of the 5' ends here to leave a gap. And then this gap structure under normal circumstances with the other allele that you might have, the homologue, would then undergo strand invasion and you'd get template switching to correct the defect. But in the case of Cas9, what we do is to offer up a DNA, single-stranded DNA template that can be used for homology-dependent repair. And so what I've shown you here, for example, let's say we're trying to knock a GFP into the coding sequence of a gene. We make a single-stranded DNA fragment that has homology to the target region at the 5' end and the 3' end. You can think of these as arms of homology if you like, but they don't have to be very long. Anywhere between 50 and 150 bases is fine. And then what we think happens is that you get pairing initially and then copying from one of these strands. This then causes strand displacement, and you will get mismatched repair enzymes that chew back either one or the other of these strands. If it chews back this strand, then you can get copying of this repair template, ligation, and then that's how the repair is affected. So, considerations for projects involving Cas9. With any project, there's going to be genetic mosaicism. Okay? There are very few exceptions we've found to this. So in practice, what this means is when somebody wants to do Cas9, they say, oh, when can I get my injected mice? You know, we usually say, not so fast. Okay? We want to give you something that's going to work. So we're going to take your G0 or first generation mice, which could be genetic mosaics. We're going to breed them and validate what you really have in the F1s is what you want, and then we'll give those mice to you. If you understand the caveats, we'll give you the G0 mice, but 9 times out of 10, people will come back to us and say, oh, could you fix this for us, please? And we go, sure, okay? So genetic mosaicism is one of the challenges. Tissue mosaicism, okay? So just because when you cut a piece of a mouse's tail off or take a few distal toe clips and analyze it molecularly, just because your alteration in the G0 mice is there, it might not be in the germline. So that's another reason why it's real important to take the F1s and to analyze those, because you know that has come through the germline. Okay, so if you want a point mutation or small deletion, etc., pros, it's relatively quick and easy, provided that the Cas9 is cutting efficiently. And that's another caveat, is that not every locus is the same. So you can design guides, and in theory, they should be able to cut, but in practice, they won't always cut. So John, to his credit, a few years ago now, decided that for every targeting, he would try and choose 2 independent guides that are very close together. By doing that, we get this sort of effect where 9 times out of 10, we'll get something successful. If you want to introduce a heterologous cDNA, this can be much faster than manipulating in ES cells. But one of the cons is that when you use these longer, what are called megamer, this is IDT's term, for a long single-stranded piece of DNA, when this is being used as a repair template, you can have minor errors involved in the repair process. So we do have to go through a few mice, but the error frequency may be no more than, say, 50% of all animals analyzed. So we can still usually get you what you need. Introducing LOXP sites, we used to do this using what's called sequential LOXP injection. Basically, we would put 1 LOXP site in, and then we generate mice, then we do IVF, and we'd put the second LOXP site in. We still have to do that sometimes, but more recently, we've been starting to do this now using megamers, and John has come up with a few clever tricks that will allow us to increase the frequency of floxing an allele, as you would say. So this is now, we think, working quite well. Okay, so we take our combination of the nucleoprotein, riboprotein complex. We'll go ahead and take our microinjected oocytes and generate mice. And we can do these sort of things, which are probably the most common things requested by clients. And we've done over 50 such projects so far. So what I'll do in the last few minutes here is just give you a sort of 10,000 foot view of how we go about doing something. So for example, here is a client that wanted to have a floxed allele of this gene target of MIB1 membrane trafficking, which is called TOM1. So the first thing that we do is we pull up the genomic structure of the locus. We look for repetitive sequences. We look at the pattern of splicing to see if there's alternate splicing around particular exons. We look at the phase of the exons to determine which ones we can remove to then generate a nonsense-mediated decay, or certainly no product being made. And then we go ahead, the royal we usually being John, to identify guides at locations where we want to cut. So we don't want to interfere with the splicing here where possible. So we'll keep these 100 bases or so away. And we'll design a template that is this sequence plus, in this case, a couple of LOXP sites. And then we'll microinject that, we'll screen the offspring and try and identify animals that have this so-called floxed allele. And what you would get back then is your sort of QC product along with your mouse would look something like this. So here's your diagram ready for publication or presentation. And here's some sequence traces showing a 5' LOXP site. Here's an intron jumping to a LOXP site, going to the intron here. And the 3' LOXP site, again, intronic to LOXP and to intron. Okay, so we won't give you something unless we're absolutely sure that it's what you want. Okay, so we really try and pride ourselves on thinking like an investigator. We don't want you to spend your valuable money and then get sunk a few years later because you end up getting the wrong thing. Here's a second example. This is an investigator who wanted to make a model of prostatic intraepithelial neoplasia, or PIN. And so what this investigator wanted to do was try and express a regulator of the cell cycle. And by doing so, he could then increase the proliferation of prostatic cells. And so he wanted to have this done, however, in a particular mouse background. And so what we worked with this investigator was to make a human SCIP2 cDNA then, which would lead to slowly deregulated cell proliferation, and to fuse this at the AUG initiator codon within the probasin gene, which is exclusively expressed at high levels in sexually mature male mice. And again, having flanking arms where we could actually integrate this thing, so 140 bases at one end and 130 at the other, to integrate it into the cognate locus. So a little bit more detail here. We do an initial screen looking for integration at the correct site using primers that are outside the regions of homology. Here's some of Shuling's work showing you this initial screening here, and the animals identified in yellow here are ones that potentially have the correct sequence. So 25 out of 74 pups born had this sequence integrated. We analyzed 10 of these and sequenced them entirely. Just over half of these had the correct sequence. 4 of them had occasional point mutations, which is something you definitely don't want. So this, in essence, gives us then about 25% of all of the positive animals being something that could be used. And once again, we will do the sequencing to demonstrate that you have the correct sequence. Here are the overlaps between the genomic sequence and the megamer that was used. Here's where the SCIP2 sequence starts, and then here's the tail end. So this is another example of the sort of what we deliver to you as the final product. Okay, so in brief summary here then, in this penultimate slide, since June, this was updated. We started doing gene deletions several years ago now. More often than not, when we want to do a gene deletion, this works really, really efficiently. Point mutations, we've done plenty of those. Conditional alleles, we are doing either by sequential LOXP insertion or by megamers now. We did try using a large double-stranded DNA to make knock-ins. Some investigators have found this to be useful, but in our experience, the efficiency is pretty low. Plus, double-stranded DNA is very toxic to pre-implantation embryos. So for everything that we do now, we pretty much always use single-stranded DNA. And with megamers, then this again is usually anywhere between 5 and 50% on average of the animals we generate. So that's nice because it means that we don't usually have to screen a lot of animals. If we make 10 or 20 animals for your project, with a bit of luck, you'll get what you need the first time around. Okay, so just to wrap up then, once again, here's our website. And again, this is probably the most important link. If you have a question, you can click on this and it'll open up in your email, and you can email this poor unfortunate individual sitting here in the front row, who will then send you something back and coordinate assistance for you. Okay, so I will stop there and happy to try and answer any questions for you. Yes?
For transgenic mice, alongside with using CRISPR-Cas9, do you also use or utilize CPF1?
Yeah, we haven't used CPF1 yet, just because we haven't. So CPF1 is a different type of CRISPR type enzyme, and it uses a different PAM site. Okay, but we found that with NGGs being the target for Cas9, we just haven't found the need yet to go to that. Some investigators that may work in model systems that are particularly AT rich might need to use that, but for the mouse, pretty much we haven't had to do that yet. If you do want it, we do have some, and we're happy to try it.
Yeah.
Yeah. Good question. Yes.
So if I would like to insert or introduce exogenous genes into mouse, and that will only express in liver, kidney, and colon, how long would that take to have that type of animal?
Okay, so the question is, if you want to introduce exogenous sequences and have them expressed in a tissue-specific manner, how long would that take? It really all depends on exactly how you want to express things. But to give you some idea, if you wanted to make a conditional allele such that you could induce through Cree expression of a coding sequence in, say, liver, kidney, whatever, then probably the way we would look at it first is to look at the sequence you want to express. And we would actually, and we've done this already for some other investigators, take an existing line of mice that have what's called a lock-stop-locks cassette driven by a common promoter. And then what we would do is retarget that mouse using your coding sequence. So we've done this for some other investigators where we've taken a GFP type mouse and knocked in a different coding sequence. And then we would go ahead and take that animal, and if the knock-in was correct by CRISPR, you could use that in combination with different Cree lines to mediate expression in different tissues. I would add that if you go look at this online resources, you'll find links to Cree databases where you can click on things and do searches for, heart-specific Cree, liver-specific Cree, kidney-specific Cree, etc. Okay. So the question was, how long would that take? Well, the annoying answer is it depends. So it really depends on what your coding sequence is like and whether we could use that approach. If we could, then it's not out of the realm of possibility that you could get F1 mice with your targeting event in there within, say, 4 to 5 months, okay, from start to finish.
6 months to a year, to be realistic.
That's my balancing influence.
It's 2 generations of mice, that's 6 months right there.
Well, 1. Let's talk about it. 6 months to a year. Okay, good question. Yes, young lady in the front.
How do people think about off-target effects?
Yes, so...
Should they sequence the genome of the entire mouse?
Yes, so this is a great question, off-target effects. Off-target effects are really an issue when it comes to working with cell lines. Yeah, because you can't breed the cell line. We have the advantage in the mouse that we can identify locations where there might be an off-target. And so John will judiciously choose guides to use that aren't found on the same chromosome. So when you backcross these animals a generation or two, then any unlinked locus that might be targeted will pretty much be removed. Where off-target effects are more of an issue is in the clinical application. So there are some very nice papers come out recently with powerful methods that will allow you to rapidly determine if there are off-target effects, say, in people that are being treated in primary tissues through gene transfer. And that's where it really is much more of a challenge.
So with the mouse experiments, would people typically go forward with a single mouse?
No, well, yes, so you get to another point. Historically, when people first started using ES cells to do homologous recombination, the gold standard in the literature was to always have the same phenotype in 2 independent mouse lines derived from 2 independent clones. And we try and do the same thing in the TMF. We try and give you a minimum of 2, ideally more. In the case of the SCIP2 targeting project, there was oodles of them, and say you should analyze these and do at least 2 independent lines and make sure you have the same effect. And that's another way of minimizing risk due to off-targeting. Okay, any other questions? All right, thanks a lot for your time.